Hey guys, welcome back. So today we're going to continue our tour through the cell by starting sort of in the interior of the cell, the nucleus, and working our way out. So last time, the last couple of lectures, we talked about the things that many different cells have in common and things that prokaryotic and eukaryotic cells do not have in common. So we spent a lot of time talking about general cellular structure. For a while, we're going to go ahead and focus on just animal and plant cells for the remainder of this slide set. Okay, so again, we're going to be starting in the center with the nucleus and sort of working our way out. And in the next couple of lecture videos, you'll be also learning about some of these other membrane structures that we'll have here. Okay, so again, getting ready to start with the nucleus, which is going to be structured relatively similarly in animals and plant cells. So here's a plant cell. Okay, you can see nuclear structure. Animal cell, we're going to see that that looks pretty similar. Okay, so we're interested in the nucleus because we are going to have our DNA here. Okay, and we're going to be at least part of the site of this concept of central dogma. Our central dogma of biology is basically, if you want to boil it down to it, transcription and translation. The eukaryotic cell's genetic instructions are there in the form of DNA, the nucleus, and it's going to be carried out by ribosomes that will function outside the nucleus. So we'll also be talking a little bit about ribosomes today. So the nucleus, again, is going to contain the DNA in our eukaryotic cell. We'll see some cases where there's some DNA in other places too. And we're going to be looking at ribosomes, which use this information from the DNA to make proteins. Kind of skipping over the RNA part of this story for now, but kind of also understanding that it's going to be there. Okay, so let's get back into nuclear structure, which again, in eukaryotic cells, is going to be structured really similarly. So we'll start with the outermost layer. Okay, so the nucleus is actually a very cool structure. Okay, so some things that we can note about it is that it actually has an outer membrane and an inner membrane. Okay, not to be confused with a lipid bilayer. Both the inner and outer membrane are composed of their own bilayer. So we've got our bilayer here. I think this is probably the inner, okay, and then the outer one, okay, with some space in between. All right, so you can see that it's got just some interesting space that's going to be connected up directly to the rough ER, which we're not going to talk about today, but we know it's coming. Okay, so we've got our inner and outer membrane, and this composes the nuclear envelope. So this will be the same envelope that dissolves when we are looking to undergo mitosis or meiosis, and then reforms when we've got new daughter cells. So this is our envelope. Okay, it's kind of amazing that that can take place given the complexity of this structure. This is going to be a microscopy image of the surface of the nuclear envelope. This is really interesting. They've used a technique where you actually see the sort of boundary at this outer edge. Still has the outer membrane, and they've kind of peeled that away in the center so that you can just see the inner membrane. So this is a pretty distinct structure. Okay, and you may have noticed that this sort of lunar-looking surface has a lot of dots in it. You can see we've got a lot of little purple structures here, okay, and this is the nuclear pore, okay, so that's exactly what it sounds like. It's a protein-lined structure that allows for an opening where things can pass in and out of the nucleus. So what kind of things might want to pass in and out of the nucleus? For one, we're going to have transcription, of course, occur within the nuclear space. That RNA needs to come out through a nuclear pore. Additionally, protein synthesis is going to occur in the cytosol. We're going to sometimes need proteins to go into the nucleus, like transcription factors and histones, which we'll be talking about a little bit later. So we'll be needing some things to go in and out of the nucleus. There are also some small molecules, things like ATP, different types of chemical energy, GGP, all of those types of things, nucleotides digested elsewhere in the cell, we may need to bring into the nucleus. So we need to have an opening. And this looks like it would just be an open door portal where anything can go through. But actually, we're not going to talk about it in class, but this is a really controlled process. Things really can only get in and out of the nuclear pore if they are supposed to go in or out. So there's a high level of control at the nuclear pore. So this is a very cool structure that we'll talk a lot about in advanced cell if you do take that. We can also see, sort of studied along, we're going to briefly talk about ribosomes, okay? And you can see they sort of line the outside, the outer membrane of the nucleus, and the connected rough endoplasmic reticulum. So we know that ribosomes are going to be responsible for protein synthesis. We'll be talking more about things like free and bound ribosomes in the next few slide sets, so you don't have to worry about that now. But just to understand their relationship in this idea of central dogma, we're going to have, again, transcription occurring in the nucleus, and then our RNA will exit through the nuclear pore and be used by ribosome to translate that message into a functional protein. Okay, so kind of understanding the relationship here and just getting an idea of where the structures are. So again, these are bound ribosomes. We'll be talking a lot more about these in the next couple of slide sets. Okay, so we can then kind of zoom in on this nuclear envelope and just lining the inner part of the nuclear envelope, we're going to have a structure called the nuclear lamina. Okay, so these are actually made of something called intermediate filaments, generally lamins. We're not talking about intermediate filaments just yet, but we will before we get to the end of chapter 6. So these filaments are designed to provide structure. So think about it like reinforcing something, like a screen can reinforce the inside of this nuclear envelope. And additionally, it actually can provide places for chromatin to attach. So we'll be learning a lot more about how chromatin is structured coming up. Okay, so we've got our outer envelope, inner envelope, some space between. We're connected to the rough ER. We've got ribosomes, nuclear pores, entering and exiting, and we've now learned more about the inner structure and this nuclear lamina. Okay, so now let's talk about a couple of other structures. All right, so we know about chromatin. This will include generally DNA and associated proteins, particularly in its sort of interphase form when it's not fully condensed, okay, just sort of spaghetti-like in the nucleus. And then we have a nucleolus, okay, so this is effectively a darker staining spot within the nucleus, okay. And so this is not actually a membrane-bound or distinct structure in the traditional sense, but this does provide a landmark. And the reason this stains more darkly than other areas of the nucleus is because it is the site of a lot of transcription. So you've got transcription occurring anywhere that genes are active, but right around this region, this is where we see just tons and tons of RNA coming off of this. And RNA will make this stain more darkly. So this is when people first started looking at cells and determining structures. And it turns out that most of the genes responsible for this high level of transcription are for what's called ribosomal RNA, which again, we're not talking too much about the ribosome today, but just understanding that this is a site of ribosomal RNA synthesis. So just a ton of it because we need a lot of ribosomes, okay? So now we've covered most of the structures in the nucleus, so it's time to really get in there and talk about chromatin. Okay, so how is this DNA arranged? So again, our chromatin is generally going to be DNA and associated proteins. And this is kind of what it looks like when the cell's just doing normal business. Okay, so we know that when it gets ready to undergo mitosis or meiosis, we'll start to condense these molecules, the DNA molecules, into what we refer to as chromosomes. So this is a highly condensed form. So this highly condensed form that we're generally used to seeing is not really how it looks in the cell most of the time. Most of the time, we're a little bit more spread out. So we're going to be talking about how the cell achieves that structure. Okay. So again, within the nucleus, we're going to really be packaging this chromatin. Okay. So everybody, I hope, is aware that DNA is a double helix. So here's our two strands, our double helix. And this is how everyone draws DNA, how it's represented. But it turns out that DNA is never just naked in a eukaryotic cell. It is always packaged with proteins. So most of the time, these proteins are going to be general sets of histones, okay? And so what we're going to start seeing is this packaging is going to help us start to compact a DNA fragment that is 2 nanometers in diameter, but really long, like 6 feet long, I think is what they say for human chromosomes, fully stretched out. How does that fit in the cell? Again, we're going to start compacting it around these proteins, okay? So let's take a second just to think about the structure that we've got here. And then on the next slide, we're going to see a little bit more detail about these histones. So histones are proteins, okay, and they are going to be composed usually in packages of 8. Again, we'll see more of this on the next slide. And this 2 nanometer double helix is just going to loop around it a couple of times. You can see there's sort of a little bit of space between each one. Eukaryotic DNA always is packaged on histone groups, okay? They're never going to be naked like this. There's always going to be histones there, okay? And then you can see, well, extra histone just to kind of staple it in place, okay? So from there, we can start to take these sort of spools. This is called beads on a string. We're starting to see that we're not quite as stretched out and long and thin, but now we're going to start getting a little bit thicker, 10 nanometers in diameter, but compacting this up lengthwise. We can continue to wrap these histone structures, the histones plus the DNA, we'll call these nucleosomes, into a more compact structure. Okay, so let's take a look at what these histones are going to look like up close. So again, histones are proteins that help pack DNA by acting as a scaffold for the DNA to loop around, as we saw on the previous image. These histones also have properties that help loosen or condense DNA, but we're going to talk about that another time, probably chapter 18. Okay, so you guys probably saw that the histones on the previous slide had little tails. That's true. I didn't draw them in all my PowerPoint skills, but those are going to help us out a little bit later. Generally speaking, there are several types of histones, but the 4 main types that we like to talk about are H2A, H2B, H3, and H4 stands for histone. 2A, histone 2B, histones 3 and 4. And we actually have 2 of each of these to create what is called an octamer. We can then go ahead and loop our DNA around twice. Yes, I know, it's not the art you hope for probably, but we saw a better image of this, again, on the previous couple of images. So the DNA loops around approximately twice and then continues on its journey, okay? So this combination of looped DNA and these histones, this histone octamer, is called a nucleosome. So when we say nucleosome, we're talking about proteins, histone group plus this DNA loop. So that's what we're looking at. Okay, so now we've had a little bit more detail about histones. Let's go back to our DNA that we're slowly condensing into a chromosome. So we started out with the 2 nanometer fiber, just naked DNA, which we now know does not exist in the eukaryotic cell. We've looped them around these histone groups to form nucleosomes, compacting this to 10 nanometer diameter, okay, called the beads on a string. To continue with that, we took our nucleosomes and we're now kind of coiling these together, like if you had a lot of spools of thread that you packed together. You can see that this is starting to get really thick and not as stretched out. This is called the 30 nanometer fiber. Okay, there's not another cute name for this. This is the 30 nanometer fiber. We can take that 30 nanometer fiber and start to sort of loop it around garden hose style and stack these loops on top of themselves with sort of a protein scaffold in the middle. Okay, so we just loop this around. But you can start to see this is our 30 nanometer fiber, and we are really just looping this around so that we're now a lot thicker, 300 nanometer territory. Okay, so this is our looped domain. Okay, that's another name for this, is the looped domain. And then these loop domains can be even further coiled up and compacted into what we understand to be a metaphase chromosome. Okay, so again, metaphase chromosomes happen only, of course, when we're undergoing metaphase. But when a cell isn't trying to divide, it's not moving day, it doesn't have to pack all its stuff up, it tends to hang out somewhere around the 30 nanometer fiber territory. So maybe a little bit looser than this, maybe a little bit tighter depending on what's going on. But this is actually the nice working range for DNA. So you can see that it is not just a double helix naked, it's all wrapped around all of these histones. Okay, so again, these histones will have properties that allow this to loosen and tighten up. But we're going to talk about that more in later chapters. For now, all we have to do is understand kind of how chromatin is structured and that it's only going to condense into visible chromosomes during mitosis. Okay, so we've talked about the DNA, we've talked about the nuclear structure, we've talked about other things in the nucleus like the nucleolus, we've talked about at least some component of ribosomal synthesis, talking about the ribosomal RNA that will be formed here, and we know that it's the site of transcription. Okay, so we'll take just a second to reconnect those ribosomes back to what's going on with the nucleus. Again, we'll learn a lot more about these later, just so that we're very sure what's going on and how they connect with our central dogma. Okay. So what we're actually looking at here is a very cool transmission electron microscopy image. We're looking at the endoplasmic reticulum, which we haven't talked about too much today, but we've seen surrounding the nucleus. So this is actually going to be the lumen, this really skinny part. I know it doesn't make sense, but that's the lumen of the ER, and then the membrane on either side. You can see lots of ribosomes just along it. Okay, these are bound ribosomes. This larger space is the cytosol between our ER, so these are going to be free ribosomes. We're going to talk about free ribosomes and bound ribosomes in the next set of slides. Okay, so our ribosome, general structure, large subunit, small subunit. We have lots of time to talk about its structure. What I want everybody to understand for now is sort of where they're positioned in the cell and that they function to produce proteins as part of this whole central dogma idea of DNA. We get copies of that by transcription, get RNA. That RNA will be translated by ribosomes into a protein. So as long as we understand that relationship, we're good for today. So once you've finished this, you should be able to generally describe the structure and function of the nuclear envelope, talking about things like the nuclear pore, these double membranes with the space between and how it's connected to the rough ER. We should understand how the nucleus is connected to protein synthesis in the cytoplasm. So specifically, again, this idea of DNA, which lives in the nucleus, get a copy that's RNA, which is then translated, okay? And we understand that the nucleolus is involved in protein synthesis because it's the site of a lot of ribosomal RNA production. That's where we're going to see high levels of transcription of ribosomal RNA. And so with that, we'll finish up, and in the next video, we'll go back to that idea of DNA, and you guys will be able to review the structure and those base pairing rules of DNA.